Now let's discuss retina cells in depth. The layers between the retina surface and the receptor cells contain several excitable cells, called bipolar, horizontal, amacrine, and ganglion cells. The vertical bipolar interneurons lie between the receptor cells and the ganglion cells, which transmit impulses to the rest of the central nervous system via axons in the optic nerve. In addition, this complex structure also contains two groups of interneurons, horizontal and amacrine cells, that function by exerting their influence in a horizontal manner by causing lateral inhibition on surrounding synaptic connections between receptor cells and bipolar cells and ganglion cells, respectively. Each eye contains approximately 126 million photoreceptors, but only 1.5 million ganglion cells. This means that there is a substantial amount of convergence of receptor and bipolar cells onto ganglion cells, but this is not uniform across the retina. At the periphery, there is a large amount of convergence, but in the region of greatest visual clarity, the fovea centralis, there is a 1-1-1 connectivity between a single cone receptor cell, a single bipolar cell, and a single ganglion cell. The fovea region has a very high density of cones and very few rods, whereas there is a more even distribution of rods and cones in the other regions of the retina. Each ganglion cell responds to changes in light intensity over a limited area of the retina, rather than to a stationary light stimulus. This limited area is called the receptive field of the cell and corresponds to the group of photoreceptors that has synaptic connections with that particular ganglion cell. Approximately half of the ganglion cells in the retina respond with a decrease in firing of their impulses when the periphery of their receptive field is stimulated by light, and increase their firing rate when the center of the receptive field is lit up. The other half increase their firing rate when the periphery is illuminated and decrease their firing rate when the central receptors are stimulated. This allows the output of the retina to signal the relative brightness and darkness of each area being stimulated within the visual field. The ganglion cells are further subdivided into two main groups, P cells and M cells. P cells receive the central parts of their receptive fields from one or possibly two, but never all three, types of color-specific cone, whereas M cells receive inputs from all types of cone. The optic nerves from the two eyes join at the base of the skull at a structure called the optic chiasma. Approximately half of each of the optic nerve fibers cross over to the contralateral side. The other half remain on the ipsilateral side and are joined by axons crossing from the other side. The axons of the ganglion cells from the temporal region of the retina of the left eye and the nasal region of the retina of the right eye proceed into the left optic tract, whereas the axons from the ganglion cells in the nasal part of the left eye and the temporal part of the right eye form the right optic tract. The neurons that make up the optic tract connect to the first relay stations in the pathway, the lateral geniculate bodies, the superior colliculus, and the protectal nucleus of the brainstem. Those fibers that synapse in the superior colliculus and the protectal nucleus are involved in visual reflexes and orientating responses. A small number of fibers also branch off at this point to synapse in the suprachiasmatic nucleus, which is concerned with the body clock and circadian rhythms within the body. However, the bulk of the neurons reach the lateral geniculate nucleus in the thalamus. Each nucleus contains 6 cellular layers, and the information from the two eyes remains separate, each group of fibers synapsing in 3 of the layers. The M ganglion cells terminate in the lower 2 layers, called magnocellular, because the cells are relatively large in these layers. Cells in the magnocellular layers are sensitive to contrast and motion, but not color. The P ganglion cells synapse in the upper 4 layers of the lateral geniculate nucleus 2 for each eye, called the parvocellular layers. These layers contain relatively small cells which transmit information about color and fine detail. The fibers from the lateral geniculate nucleus fan backwards and upwards in a bundle, called the optic radiation, through the parietal and temporal lobes to an area of the cerebral cortex called the primary visual cortex. So that was all for today's topic. For more such videos, keep following sqadia.com.